Very much. Sorry about that delay. So this talk is about early innate immunity in the upper respiratory tract to SARS-CoV-2 infection. This is a topic that has become very important because the infection with the virus really the early phase of the viral infection is rather important, as has been shown by people like Shane Crotty and Alejandro Sete and so on. And this is a slide showing the general consensus that has been achieved by most people working on the immunology of SARS-2. And that is that you have an early innate immune response followed by an adaptive immune response, which comes out more slowly. And if you have a good enough early immune response, which could be primarily an innate immune response with some elements of adaptive immunity coming in, then the viral load in the upper respiratory tract disappears. And therefore, you do not get to the progression towards a serious COVID-19, where you get infection of the lungs, because the virus then gets into the lungs, establishes itself there, and causes very severe illness or death. So the key point of all our efforts in vaccination and so on is to try and reduce the viral load early on in the upper respiratory tract. You have essentially trained adaptive immunity coming into play, involving T cells, T cell mediated immunity, and antibodies. And then innate immunity, which is an inbuilt function of the upper respiratory tract. So firstly, I'd like to look at the concept of nasal air conditioning. And this, I think, probably has been fairly underestimated. And this doesn't involve any kind of specific immune response, but essentially inspired air, which is typically in the summer in the temperate climates, is warmed to about 34 degrees, 33 degrees in the nasal cavity, and then humidified to about 90% relative humidity. And this then increases as the air goes further down to about 37 degrees, and then humidity of 100%, which is essential for lung function. And there is a subcapillary, sub-epithelial capillary network in the nasal cavity and the nasopharynx, which actually brings about this warming of air, particularly in the nasal cavity. And this warming of air, or the nasal air conditioning and its humidification, is essential for optimal mucosal barrier function. It's important for mucociliary clearance of the virus. It's important for, as we shall see, for the other types of innate immune responses. And details have been published already, and you can read about it in the papers listed at the bottom of the slide. Now, one thing that's becoming very clear is that SARS-CoV-2 really infects the nasal epithelium first, rather than the nasopharynx, which of course it infects later. And that the virus replicates better at lower temperatures in the nasal epithelium. And it's been fairly well established now, I think, that the weaker induction of type 1 and type 3 interferons as a result of the viral infection is weaker at lower temperatures. And the interferons that are induced in this way produce various types of what are called interferon-stimulated genes, products, proteins that particularly do the downstream work of keep making the epithelium resistant to further viral infection and getting rid of the virus from the epithelium. Now, we postulated earlier on that selection by respiratory virus may be an important feature of optimal nasal conditioning in temperate climates. And there are differences in nasal structure and nasal air conditioning ability, we believe. Certainly nasal structure and air conditioning ability hasn't been studied all that well between tropical and temperate zone populations. And it's been postulated that this increases the susceptibility to COVID-19 infections, unfortunately, for people of tropical origin living in temperate climates. And therefore, we have to take better, precautions and care with such populations. Now, this has been the increased susceptibility of migrants from tropical areas living in countries like countries of Western Europe and the US has been put down to socioeconomic factors. And I'm sure that they do play a part, but we also have to look at the opposite, and that is there may be people from tropical areas are not necessarily, and they don't have the need to deal with respiratory viruses in cold temperatures, as one has to in Europe. And this may probably have thousands of years ago exerted a significant selective pressure in European countries. Okay, so nasal conditioning, the need to warm air is important, and it may not work well in winter. And we all know that influenza and SARS-CoV-2 does tend to be more infective in winter. And it's also been a very old human tradition to wear scarves to cover your nose to keep your nose warm in temperate climates and even in tropical climates in hill countries. So this is an old process that unfortunately we are having to face with COVID-19. And just to go on a little bit further, quite independently, the Chicago group published last year this rather interesting finding, which shows that there is a difference in interferon responses in people, and they only looked at African genetic ancestry and European genetic ancestry. And what they showed was the interferon response is much greater in European populations. And this is a rough score. I won't go into the details of that. And this is looking at monocytes and a number of other peripheral blood cells rather than the upper respiratory tract. The interferon response is much greater in European populations. And you may well be, and this is in response to influenza A infection of cells rather than the upper respiratory tract. And you may well relate this to possibly something happening in the upper respiratory tract to prevent SARS-CoV-2 infection. Now, moving on, the immune response, the molecular basis of the immune response process has been much better studied in influenza virus infections than in SARS-2, but it's advancing very, very rapidly. Influenza virus is another RNA virus. It's a negative strand RNA virus, so it differs from SARS-2 in having a nuclear phase as well. The replication transcription takes place in the nucleus as well. So when the virus, an RNA virus gets into the cell, it's recognized by specific pattern recognition molecules, which then activate an innate immune response initially. And these include molecules like toll-like receptors, which are on the membrane or in endosomes. And then there are also cytoplasmic molecules like what's called RIG1 and MDA5, which are fancy original names. And these essentially produce a number of interferon regulatory factors, IRFs, which stimulate the production of type 1 and type 3 interferons, the synthesis in the nucleus. And then in a complicated process, these type 1 and type 3 interferons are able to bind to receptors either on the same virus-infected cell or more relevantly, perhaps, neighboring epithelial cells. And then through a signaling process that involves other interferon regulatory factors, cause the synthesis of interferon-stimulated genes, which do the work of actually preventing viral infection, replication, and killing the infected cell off. So these are the interferons. There are lots of them, interferon-stimulated genes, which are important for preventing viral replication. Also, you have a branching pathway which stimulates the production of caspases, and these are involved in production of inflammatory cytokines, IL-1, and so on, IL-18, which are then involved in kicking off or kickstarting the adaptive immune response. They bring in inflammation, they activate antigen presentation, and bring in T cells and B cells in the lymphoid tissue. So that is the long and the short of the response to an RNA virus. And these can be summarized here as well. Essentially, what we find is that there's an increasing amount of evidence that children have a lower risk of severe COVID-19. And this is being related by several authors, several pieces of research, to earlier and better innate and adaptive immune responses in the upper respiratory tract. Early induction of interferon-stimulated genes and inflammatory pathways in the upper respiratory tract is associated with reduced SARS-2 replication and decreased COVID-19 severity. Lots of bits of evidence, so the evidence from studies of children and the increasing numbers of in vivo studies in the upper respiratory tract support these two assertions. We also find that there's defects in certain pathways that are involved in producing antiviral immunity in the upper respiratory tract are associated with increased susceptibility to COVID-19. So defects in TLR toll-like receptor 3 or interferon regulatory factor 7 genes are associated with far greater susceptibility to COVID-19. And also some individuals who have antibodies to type 1 interferon, naturally formed autoantibodies due to various reasons, are also more susceptible to COVID-19. And people who have defects in antiviral interferon-stimulated genes are also more susceptible to COVID-19. And these include, for example, I'll just give one the oligoadenylate synthase gene, which produces a rather interesting molecule, 2' 5' oligoadenylate, which stimulates a ribonuclease, which then serves to stop viral multiplication and also cellular multiplication, cellular transcription and active gene protein synthesis in the infected cells. So there's quite a bit of evidence now that building up onto the importance of innate immunity, early innate immunity, that's not involving antibodies or T cells in susceptibility to COVID-19 and early SARS-2 replication inhibition. And this is a very complicated slide, which you don't need to, which I won't go into details of, but essentially it summarizes, and this is work done by Galani et al., summarizes that defects in these genes like toll-like receptor 7 and also signaling molecules, including the interferon A receptor, which is on the cell surface, genetic defects in these increase susceptibility to COVID-19. And antibodies to interferon, type 1 interferons also do this. So basically, this is just recapitulating what I said earlier, and also OAS, oligoadenylate synthase genes, defects, natural inherited gene defects are involved in greater susceptibility to infection. So we think that this is also these are also playing a role in the upper respiratory tract. Additionally, then we have a number of proteins coded for by the SARS genome that also inhibit innate immune signaling. And this seems to be something that the virus has accumulated in its evolutionary history, starting from back coronaviruses or whatever, to interfere with innate immune signaling and therefore enabling the virus to become more infective. So in summary, this is a slide which just basically summarizes everything. So the genetic factors that are involved are nasal air conditioning ability, and that involves the upper respiratory tract, the nasal and nasopharynx in particular, and also antibodies and immune effector cells that come into play, which the nasal epithelium also has lymphoid tissue. There are also tonsils and adenoids and other lymphoid aggregates, which is where the adaptive immune system starts off. These are where the inflammation, infection, and signaling causes T cells and B cells to become activated. And these may, after vaccination, for example, come to reside in the nasal epithelium, giving greater immunity. So host gender. The other factor that I didn't mention is age and gender. So there is good evidence that nasal conditioning ability decreases with age, and this will reduce innate immune responses in the upper respiratory tract. And there's some influence possibly of gender, not so well established. There are some effects like pollutants and obviously virus variants, which will affect the innate immune responses, the early immune responses, and temperature. I mean, if the winter temperatures drop down to say 4 degrees, and inhaling air at 0 degrees or even below that, and that's generally dry air, the innate barrier immunity of the mucous system and the innate early immune responses are drastically reduced, and that will reduce the effectiveness of the adaptive immunity. So that I hope has given you some indication of the complexity of the genetic features that govern early immunity to SARS-CoV-2 and susceptibility to infection. Details are in this publication, which came out recently. Thank you very much. I'm happy to answer any questions.